So we have been discussing on the metabolism of cholesterol, and we have completed the de novo synthesis of cholesterol, which is mostly happening in the cytoplasm, and the remaining modifications which are happening in the microsomes or the endoplasmic reticulum. Right. So cholesterol is a 27 carbon compound synthesized from the 2 carbon compound acetyl CoA, right, and through a series of reactions around with the help of 30 different enzymes. The cholesterol, the one of the most complicated structured compound, cholesterol, is synthesized, right? And now we are moving on to the degradation of cholesterol. So what happens after its lifetime is attained, or what happens when it is subjected for breakdown, degradation? What is the fate of cholesterol in the body? During the functions, when we were discussing on the functions of cholesterol, we have discussed that, we have told that the cholesterol is one of the most important compounds which is responsible for synthesizing the fat soluble vitamin D. Then it is responsible for the compounds which are responsible for carrying out the emulsification reactions, that are bile acids, that is the bile acids. Then it is again responsible for the synthesis of adrenal cortical hormones, or otherwise known as steroid hormones and all, right? So all of these biologically important substances are synthesized from cholesterol, and for the synthesis this cholesterol will be subjected for degradation. For example, again one of the most important compounds responsible for the synthesis, responsible for the emulsification reactions, the bile acids. And these bile acids, they are synthesized in the hepatocytes. The bile acids are synthesized from cholesterol in the hepatocytes, and they are stored in the gallbladder and secreted into the intestine through the biliary canaliculi or bile duct. Right. So, what is the function of these bile acids? These bile acids carries out the emulsification, or it enables the digestion of lipids, the dietary lipids. Okay, so in order to carry out the digestion of lipids in a smooth manner, the amphipathic molecules are formed, are needed, and one of the molecules is the bile acids, and these bile acids are again synthesized from the cholesterol itself. And this formation of formation of bile acids from the cholesterol happens in the liver cells or in the hepatocytes. But as cholesterol is a 27 carbon compound, while bile acids are 24 carbon compounds, right? The bile acids are again divided into two different types, the primary bile acids and the secondary bile acids. So all of them are synthesized from the cholesterol. So the bile acids which are directly synthesized from the cholesterol are known as primary bile acids, and they are found in the hepatocytes. And once they are synthesized, they will be moved towards gallbladder, where they are stored. And once they are required, they will be secreted into the intestine through the biliary canaliculi. So how is the cholesterol degraded to give rise to bile acids? That we are going to study today. So these are the fates of cholesterol in the body. Main functions of the cholesterol, that is, they undergoes degradative reactions or catabolic reactions in the humans to give physiologically important byproducts like bile salts, bile acids, steroid hormones, and vitamin D. So again, among them, the most important compounds are the bile acids and the bile salts. So what is happening to cholesterol so that they get converted into the bile acids? So about half of the cholesterol in the body is ultimately converted into bile acids and bile salts, right? So, bile salts are nothing but the sodium and potassium salts of bile acids. So what are bile acids? Bile acids again fall into two categories, the primary bile acids and the secondary bile acids. The primary bile acids are the cholic acid and cholic acid. So they are the primary bile acids, the cholic acid and the cholic acids. Why these bile acids are important for the body? Because they are responsible for carrying out the conjugation of various substances, right? So the bile acids again are not found in the free form. The cholic acid and cholic acids are not found in the free form. They will be conjugated with the amino acid glycine or taurine to give glycocholic acid, glycokenodeoxycholic acid. Similarly, taurocholic acid and taurokenodeoxycholic acid, etc. So how are they synthesized from cholesterol? That we are going to study. So cholesterol will be first converted into a compound known as 7-alpha hydroxycholesterol. We know that the cholesterol contains, the structure of cholesterol contains an OH group on the third position, right? So that is the structure of cholesterol. That is cholesterol having 4 ring system, namely A, B, C, and D, of which D is a pentane molecule that is having only 5-membered ring system, while the remaining the 3-ring systems are 6-membered ring systems. And among again the ring B contains a double bond in between the 5th and the 6th carbon atom, right? And the D ring is having an isoprenar side chain which is having 8 carbon atoms. Apart from that, there are 2 more side chains which are one located on the 10th and the other one on the 18th position. That's a methyl group, right? So this forms the overall structure of cholesterol. And what happens during its degradation into bile acids? The first step to happen is the hydroxylation reaction of cholesterol molecule on the 7th position. That is, an OH group will be attached onto the 7th position of cholesterol by the enzyme 7-alpha hydroxylase. And this step forms the rate limiting step of the bile acid synthesis or the catabolism of cholesterol. So whether the catabolism of cholesterol to yield bile acids or bile salt is determined by the enzyme 7-alpha hydroxylase. So it's a microsomal hydroxylase system. It's a monooxygenase system, and the reaction require what molecular oxygen. Monooxygenase anesite require molecular oxygen, and in presence of the reducing equivalent NADPH, then vitamin C, cytochrome P450, etc. So in presence of all these coenzymes and cofactors, see, the hydroxylation reaction will be happening on the 7th position. We know that this is the 7th carbon atom of cholesterol molecule, and this 7th carbon atom will be having a side chain, that is OH group, by the enzyme 7-alpha hydroxylase in presence of NADPH and molecular oxygen. So that NADP and water is formed, and the cholesterol is converted into 7-alpha hydroxy cholesterol. Now what is happening? Majority of the 7-alpha hydroxy cholesterol will be converted into cholic acid, which then further undergoes conjugation with glycine to form glycocholic acid or taurine to form taurocholic acid. So these are the primary bile acids: glycocholic acid, taurocholic acid, glycoquinodeoxycholic acid, and tauroquinodeoxycholic acids. These are the primary bile acids. And from the primary bile acids, the secondary bile acids will be synthesized. That will come to learn in the following slides. So some of the 7-alpha hydroxycholesterol is converted into quinodeoxycholic acid. And what is happening? This combined with glycine to form glycoquinodeoxy and tauroquinodeoxycholic acid. So these are the cholic acid and quinodeoxycholic acid. They are synthesized in the liver from cholesterol. So this is the reaction happening. So cholesterol is acted upon by the rate limiting enzyme 7-alpha hydroxylase in presence of NADPH, molecular oxygen, vitamin C, cytochrome P450. All in presence of all these compounds, 7-alpha hydroxylase converts cholesterol to 7-hydroxy cholesterol or 7-alpha hydroxy cholesterol. And this 7-alpha hydroxy cholesterol, what happens? It is converted into cholic acid. So what is the change happening during the conversion of 7-alpha hydroxycholesterol to cholic acid? It will be converted. That is, we know that cholesterol is a 27 carbon compound. Now, this 27 carbon compound then undergoes the removal of 3 carbon atoms. We know that the bile acids are 24 carbon and the cholesterol is 27. So 27 minus 24, 3 carbon. So the 3 carbon atoms from our isoprenoid side chain. We have got an isoprenoid side chain on the D ring. So from the D ring, 3 carbon atoms, that is the 25, 26, 27. 25th, 26th and 27th carbon atom will be removed in the form of propionyl CoA. We know that where is this? Yeah, this is a cholesterol structure, right? So this is the 25th carbon atom, 26th and the 27th carbon atom. So these 3 carbon atoms will be removed in the form of propionyl CoA. So that now the 27 carbon cholesterol or 7-alpha hydroxy cholesterol is converted into cholic acid. Right, so that is how the cholic acid is synthesized. And in addition, one more reaction is happening. There is an hydroxylation in one more position, that is the 12th carbon atom. So 7, 8, 9, 10, 10, then 11, 12. This is the 12th position. This is the 12th position. So on this 12th position, one more hydroxylation reaction happens. So in presence of 12-alpha hydroxylase enzyme, again there is one more molecule of NADPH and molecular oxygen. Then what happens? There are 2 events happening. One is the addition of a hydroxyl group on the 12th position, and the other is the removal of the 3 carbon compound in the form of propionyl CoA. So that 7-alpha hydroxycholesterol is converted into collyl-CoA. Collyl-CoA, right? So two molecules of CoA ACH also comes, so that now it's not hydroxycholesterol. It is converted into collyl-CoA. Collyl, that is ഇവിടെ മൂന്ന് കാർബൺ ആറ്റംസ് റിമൂവ് ചെയ്തു. അപ്പൊ ഇവിടെ CH2 വന്നു. C ഡബിൾ ബോണ്ട് SCOA ആയിട്ട് മാറും. സോ ദാറ്റ് ഇസ് കൊളൈൽ കോയെ. എന്തിനാണ് കോയ ആയി മാറുന്നത് വെച്ച് കഴിഞ്ഞാൽ it is in the next step it is getting conjugated with glycine. So conjugation cannot happen in an easy way. So first this compound has to be converted into a highly reactive species. That is why cholic acid is converted to collyl-CoA. Again, propionic acid is not released in the form of propionic acid, it's in the form of propionyl-CoA. Clear. So that is how the two CoA ACH reacts. So when 7-alpha hydroxycholesterol is converted into collyl-CoA, what all things happens? 12-alpha hydroxylase introduces an hydroxy group on the 12th position of cholesterol. Then a 3 carbon unit is removed in the form of propionyl-CoA. So for the propionic acid to get removed in the form of propionyl-CoA, we require one molecule of CoA ACH. Again, cholic acid, in the next step, what it will get conjugated with glycine or taurine to give glycocholic acid or taurocholic acid. So for the conjugation reaction to happen, cholic acid is converted to collyl-CoA, right? So 7-alpha hydroxycholesterol is acted upon by 12-alpha hydroxylase enzyme, so that propionyl-CoA is released, and also 7-alpha hydroxycholesterol is converted into collyl-CoA, right? Now what happens? This collyl-CoA is undergoing conjugation to give glycocholic acid, right? Glycocholic acid and taurocholic acid, right? So that is how this is one of the primary bile acids. Now what is happening? The same 7-alpha hydroxycholesterol will be converted into quinodeoxycholic acid. In case of conversion of 7-alpha hydroxycholesterol to quinodeoxycholic acid, there is nothing happening, only the removal of propionyl-CoA is taking place. Okay, there is no introduction of a hydroxyl group. So 7-alpha hydroxycholesterol simply undergoes a removal of 3 carbon units in the form of propionyl-CoA in the presence of one molecule of NADPH, one molecule of molecular oxygen, and there is introduction of 2 molecules of CoA acid, so that 7-alpha hydroxycholesterol is converted into quinodeoxycollyl-CoA, and there is a removal of propionic acid in the form of propionyl-CoA, right? So this is how the primary bile acids are formed. Now quinodeoxycollyl-CoA also undergoes conjugation with glycine or taurine to give tauro or glycoquinodeoxycholic acid, right? So this is how the primary bile acids are synthesized. So the primary bile acids are mainly cholic acid and quinodeoxycholic acid, which undergoes conjugation with glycine and conjugate so that they are made miscible, right? So the cholic acid and quinodeoxycholic acid are converted into their conjugated form, because they are never found in the free form. They are always found in the conjugated form, and the conjugated form are the glycocholic acid, taurocholic acid, glycoquinodeoxycholic acid, and tauroquinodeoxycholic acid. So these are the primary bile acids, and from the bile acids, from these primary, all of these are synthesized in the liver. Now what happens? The secondary bile acids has to be synthesized. So how are the secondary bile acids synthesized? The secondary bile acids are synthesized. They are only this conjugated form are released from the liver, and once they are released from the liver, they enters into the they are secreted into the bile, and they will be whenever they are required, they will be undergoing salt formation with sodium or potassium. So sodium glycocolate or potassium glycocolate, sodium taurocholate, sodium glycocolate, so sodium glycoquinodeoxycolate, potassium tauroquinodeoxycolate, etc. So they are always released again, conjugated, and again they are also released in the form of salt. They exist in the form of sodium or potassium salts of bile acids. So that is how the primary bile acids are synthesized and secreted into the bile. Now, what are the secondary bile acids? The primary bile acids thus formed, they undergoes modification in the intestine. So once they are synthesized in the liver, what happens? They undergoes modification. They will be undergoing conjugation and they forms a salt form, and they will be moving into the intestine. And in the intestinal cells, or the intestinal bacteria comes and converts this into their deoxy form.